the permeability really is a reflection of the channels that are opened up. That's going to be important not only for the resting membrane potential, but also thinking further into the action potential. The second piece is thinking about the sodium-potassium pump. So the sodium-potassium pump, the direction of its ionic movement, it's moving 3 sodiums out, 2 potassiums in. So automatically more positive charge is leaving the cell, and so there's more negative charge left behind, right? The other piece is thinking about the different permeability for potassium. So the fact that there are more potassium channels, leak channels that is, so the resting membrane potential will look more closely like potassium's equilibrium potential, simply because there are more channels for potassium to move. And so potassium is going to be a little bit more at home in terms of the resting membrane potential value, right? And then we want to think about combining the direction and the magnitude of our individual forces acting on both sodium and potassium. So our chemical driving force for sodium is into the cell. Our electrical driving force for sodium is also into the cell. And so we combine those two. And so sodium always has a strong ECDF in a direction into the cell. For potassium, we have more channels, but potassium has a CDF moving out of the cell and an EDF moving back into the cell. So potassium has opposite movement in terms of its individual forces. So combining those together automatically means that it's a smaller magnitude, because we have to subtract the weaker force from the bigger force. So we subtract the EDF from the CDF, which results in 24, right? That's going to be the magnitude, and the overall direction being out of the cell. Therefore, potassium moves out of the cell with a smaller or weaker force or weaker magnitude. And then because it has more channels to leak out of the cell, it's going to move out of the cell. And so those two movements, sodium moving strongly into the cell, potassium moving weakly out of the cell, those two forces are being balanced out with the movement of both of these ions by the sodium-potassium pump. And then we arrive at the steady state. So this is just explaining why the neuron at rest has a membrane potential of negative 70 millivolts. Okay, so definitely recall all of these details. You'll see questions on some of the factual components here, right? Knowing that permeability for potassium is greater, knowing the direction of movement for sodium-potassium pump, knowing what the ECDF for both of these ions look like, knowing the resting membrane potential value, but also more of the conceptual things, understanding how we get to this number, how we have negative charge inside the cell because of the abundance of positive charge that is leaving, all right? So this is going to definitely be an important concept to go over for the exam. I'm going to move on to the action potential. All right. The other important concept to go over for the exam is the action potential. And again, factual things you want to remember are the ions that are moving for each phase of the action potential. So you definitely need to know what's happening from negative 70 to negative 55. We talked about this being the graded potentials arriving on the axon soma or cell body, and then determining if that was strong enough to get to negative 55. So the green curve here are the graded potentials. And more recently, we talked about the fact that those can be IPSBs or EPSBs, depending on what type of channels open up. and depending on whether it's a fast or slow response. All right. The blue part of the curve, which is the upstroke of the action potential, the depolarization phase, that is owed to sodium movement. So you definitely want to recall that sodium is moving here, but also think about the mechanism by which sodium is moving. Sodium is moving because at -55, the voltage gated channels are activated to open up. So the activation of the voltage gated channels means that we go from a state at rest where the channels were inactivated in that they were closed and incapable of opening. And now we're in a state going up to +30 where the channels are open. Both the activation gate and the inactivation gate is open. Permeability for sodium is high. Sodium is freely rushing into the cell according to its strong ECDF. And then again, because the inactivation gates are going to be deactivated after 1 millisecond, but also at +30, then that sodium movement is halted. And at the exact same time, potassium movement can begin after that 1 millisecond time delay. So the 1 millisecond is syncing these two events, the inactivation gates on sodium channels closing and the potassium channels opening up after 1 millisecond. And so the repolarization phase of the action potential is going to be owed to the potassium moving because of its channels being opened up. And then the membrane potential here is becoming more negative. It's going to go lower than rest. And again, understand the concept that potassium is moving to get closer to its equilibrium potential, which is way down at -94. So it's going to go past the resting membrane potential, and then eventually, as more of those potassium channels close, right, then we're going to see a correction in that negative membrane potential change. And then in the purple curve, the sodium-potassium pumps, along with the closure of all potassium channels, is what brings the membrane potential back to rest. All right. And so we want to also include here our refractory periods. So the period immediately surrounding the depolarization and repolarization, it's not possible to fire another action potential, at least not on this specific segment of the neuron. Remember, we have adjacent segments where the action potential is traveling down the length of the neuron, but for this specific segment in question, that segment will be in its absolute refractory period, and then the proximal segment, the segment before, that will also be in its refractory period. So it's impossible for the action potential to move backward. It's always going to be moving forward. All right. And then that relative refractory period will be the phase not ultimately, but immediately following the repolarization phase. So basically in the overshooting of the resting membrane potential all the way until we get back to rest, here is where our channels are closed, but they are capable of opening. All right, so it's possible to fire another action potential, but it's going to be very difficult. It's going to require a much stronger, greater potential simply because potassium channels are still trying to close. So the membrane potential is still more negative than it is at rest. All right, so these are all important concepts to go over. Again, some of the more factual things to remember, things like -55, that's a number you want to remember. +30 is a number you want to remember. The 1 millisecond time delay. You also want to think about the different channels and their gating mechanisms. Those are more factual components to go over. And then also think about the conceptual things that are, as far as where the absolute refractory period is and why, where the relative refractory period is and why as well. All right. Okay, the last thing that I want to go over in the interest of time here is basically the synapse and the events of the synapse. So let's go ahead and review that. All right, so let's look at the details as far as what's happening at the synapse. Also go over as a part of this, the difference between an electrical synapse and some of again, factual things about the electrical synapse. It's faster, it's bidirectional, it's less common or less abundant throughout the body. What else did we say about electrical synapses? They are immediate, right? So it's going to be using the gap junctions that are located between two cells. So definitely go over some of the more factual things about the electrical synapse. And then more of the emphasis was on the chemical synapse, but know how they differ. So the chemical synapse, we said, happens at a neuron to another type of cell or another type of neuron. And basically, here we're not just going from one cell to the other really quickly. Here we have to transfer that energy from electrical, which is the state moving down the axon, the action potential moving down the axon. We go from electrical energy that then stimulates chemical energy. So when the action potential arrives at the axon terminal, voltage-gated calcium channels open up. Calcium rushes into the axon terminal, and calcium is going to stimulate the exocytosis of these synaptic vesicles. And within the synaptic vesicles, we have these neurotransmitters. So these get dumped into the junction by exocytosis. And remember, we talked about that millisecond delay. So that 0.5 to 5 millisecond delay happens because it takes time for calcium to enter the axon terminal, calcium to stimulate the movement or the docking of these vesicles close to the axon terminal membrane, and then actual exocytosis to occur. So those are the activities or events that cause the delay. The delay is not caused by the time that it takes for the neurotransmitters to cross the junction, right? So that is not what causes the delay. Instead, it's really the exocytosis process. Now, the neurotransmitters will bind to the receptors, and here we can think about adding in the type of cellular, the type of receptor here. So it can be a fast receptor where the receptor is a channel. So when this neurotransmitter binds to the receptor, channel opens up. That's an ionotropic receptor. That's a fast receptor. And so ions will start to move. A cellular response will happen right away. The other option is the metabolotropic receptor, which is a slow receptor. It's coupled to a G protein. And so when this neurotransmitter binds to a metabolotropic receptor, that's going to result in a part of that G protein activating another enzyme, and then second messenger being generated, and then some type of cellular response can happen downstream. So essentially, a slow response, our metabolotropic receptor, has several steps in the way. It's kind of like a cascade of events that has to happen, which are ultimately triggered by that G protein and the alpha subunit on that receptor. Now, if the signal has been sent and we have to, we need to cease the transmission here, we have to clear the junction of any excess neurotransmitter. And so we talk about three options for clearing the junction. The first option is that we degrade any excess neurotransmitter with the enzyme on the membrane. The name of this enzyme was acetylcholinesterase, simply because most of the times acetylcholine is the neurotransmitter in question. There can be other neurotransmitters, but for the most part, acetylcholinesterase will degrade excess acetylcholine, and that will help to clear the junction. The second option was the reuptake molecule. So the reuptake molecule is actually on the side of the axon terminal. So this is going to bring that excess neurotransmitter back into the axon terminal, repackage it into a new axon terminal, synaptic vesicle, and then we can basically recycle that excess neurotransmitter. And then the third option was basically it diffuses away from the junction, and that will help to clear the junction as well. So all three of these things, it's not either or, all three of these things are happening simultaneously, and they're all helping to clear the junction of any excess neurotransmitter. All right.